Let's talk a little bit about protein structure. Now, every protein, whether it's hemoglobin or keratin or insulin, has the same old backbone of carbon and nitrogen atoms held together by peptide bonds. And so, if you remember from our previous video, I told you it always goes nitrogen, carbon, carbonyl, nitrogen, carbon, carbonyl, etc. And I said if you draw a skeletal structure, any R group that's pointing up should be on a wedge, and any R group that's pointing down should be on a dash, like that. Now, the difference in proteins is the variation in the length of the backbone and the sequence of the side chains or the R groups that are attached to the backbone. Now, the individual order in which the amino acid residues are linked together in a protein is what we call primary structure. So, primary protein structure, the sequence of amino acids. So, let's say you have a glycine, then an alanine, then a tryptophan, then a phenylalanine. That order that they come in, that's what our primary structure is. And each different protein in a biological organism has a unique sequence of amino acid residues. It's this sequence that causes a protein chain to be able to fold and curl into the distinctive shape that enables the protein to function properly. A protein molecule that loses that three-dimensional shape, its characteristic three-dimensional shape, we say that's denatured, and denatured proteins can no longer carry out their biological function. If you've ever reached into the oven before to grab maybe a hot cookie sheet or something and your skin touches it by accident and you see how your skin changes color immediately, that would be the proteins in your skin denaturing very rapidly. And so biochemists have devised techniques for finding the order in which the residues are linked together in protein chains. And a few of the many proteins whose primary structures are known are shown here in this table. And you can see that they vary from something like insulin, which is found in the pancreas and controls metabolism of carbohydrates, has 51 amino acids. Then if we look at something like human growth hormone, it has 191. But if you look at something like gamma globulin, which makes up antibodies in the immune system, it has over 1,300 different amino acids. And so now you can see why we like to use the three-letter and the one-letter abbreviations because it just speeds things up so much. And so you can see we have this peptide shown here, okay, where we have the N-terminal or N-terminus, which is a serine, and then we have the C-terminal or C-terminus, which is a phenylalanine. But each one of those circles in there represents a different amino acid linked via a peptide bond or a peptide linkage, and you can see the abbreviations of each one of those amino acids in there. So the order in which those amino acids are listed in this peptide is what we call the primary structure. If the only structural characteristics of proteins were their amino acid sequences, which is the primary structure, then all protein molecules would just consist of long chains arranged in random fashion. However, protein chains fold and become aligned in such a way that certain orderly patterns result. These orderly patterns are what we refer to as secondary structure, and they result from hydrogen bonding. Now, there's two types of secondary structure. We can have what's called an alpha helix and a beta pleated sheet. Now, the alpha helix, back in 1951, Linus Pauling and Robert Corey suggested that proteins could exist in this helical shape, kind of like the helix in the double helix in DNA, or you think about a spiral staircase. And that alpha helix is a form in which the single protein chain twists so that it looks like a coiled helical spring. Now, the chain is held together in the helical shape by all of these different hydrogen bonds between carbonyl oxygens and amide hydrogens in adjacent turns of the helical backbone. And so you can see the hydrogen bonds, shown here by these dots. And this is a more simplistic representation of it here, but you see carbonyl, then you see the hydrogen bond to a or a hydrogen that's attached to a nitrogen, right? You see it repeated here and here. And when you put them all together, that's what causes this helical shape, this spring-like shape. So The carbonyl group of each amino acid residue is hydrogen bonded to the amide hydrogen of the amino acid. It turns out that there's 4 amino acid residues away. Thus, all the amide groups in the helix are hydrogen bonded. The protein backbone forms the coil, and the side chains, or the R groups, they're going to be extended outward from the coil. And so you can see the R groups in this representation here are kind of on the outside of the coil. They're not shown in here, but they would be sticking off, something like that. Now, 6 years after Pauling and Corey proposed the alpha helix, its presence in proteins was detected. And since that time, it's been found that the amount of alpha helical content is quite variable in proteins. In some, it's the major structural component, whereas in others, a random coil predominates and there is little or no alpha helix coiling. So proteins like keratin, which is found in hair, or myosin, which is found in muscles, or epidermin found in skin, and fibrin found in blood clots are all examples of fibrous proteins. And in these proteins, 2 or more helices interact to create a coiled coil, which is like a cable, if you've ever seen a piece of metal cable. And these cables make up bundles of fibers that tend to lend strength to the tissue in which they're found. Another common type of secondary structure found in proteins is what we call the beta pleated sheet. Okay. It results when several protein chains lie side by side and are held in position by hydrogen bonds between carbonyl oxygens of one chain and the amide hydrogens of another chain. If you think about corrugated cardboard, it kind of looks like the corrugation in cardboard. And we don't go into gross detail about beta pleated sheets, but you can also have parallel and anti-parallel hydrogen bonding, which again is beyond the scope of our course. The main thing is that you remember that primary structure is the individual amide linkages between the amino acid residues. And secondary structure, there's 2 types. There's alpha helices and beta pleated sheets. Tertiary structure, the next higher level of complexity in protein structure, refers to the bending and folding of a protein into a specific three-dimensional shape. This bending and folding might seem rather disorganized or random, but nevertheless, it results in a favored arrangement for a given protein. The tertiary structure results from interactions between the side chains, or the R groups, of individual amino acid residues. And when it comes to the R group interactions, there's 4 types that you need to know. The first one is a disulfide bridge, and we actually saw that in a previous video, where a linkage can form between 2 cysteine residues that are close to each other in the same chain, or between cysteine residues, and they form a disulfide bond. Okay. The next one is a salt bridge. And these interactions are the result of ionic bonds that form between the ionized side chain of an amino acid, which has a negative charge, and the side chain of a basic amino acid, which has a positive charge, right? Positive, negative, right? Salts, cations, anions. So that's would be a salt bridge. Next, we have hydrogen bonds, and you should be relatively familiar with hydrogen bonds by now. Hydrogen bonds can form between a variety of different side chains, especially those that possess like a hydroxyl group or an amino group or an amide or something like that. So hydrogen bonds. Now, one possible interaction between the hydroxyl groups of 2 serine residues is shown here. We saw before that hydrogen bonding also determines the secondary structure of proteins. And the distinction is that the tertiary hydrogen bonding occurs between R groups in tertiary structure, whereas it occurs between the backbone of carbonyls and nitrogen hydrogens in secondary structure. All right, and lastly, we have the hydrophobic interaction, and these result when nonpolar groups are either attracted to one another or forced together by their mutual repulsion of aqueous solvent. So this type of interaction is common between nonpolar R groups like phenyl groups or phenylalanine residues, and this is shown in the compact structure of some proteins in aqueous solutions. And so that's what's being represented here in figure 12.14. And this shape results because the polar groups are pointed outwards towards the aqueous solvent, whereas the nonpolar groups are pointed inward away from the water molecules. And this type of interaction is weaker than the other three because it's based off of London dispersion forces, but it usually acts over a large surface areas so that the net effect is an interaction strong enough to stabilize the tertiary structure. And so if we take a look at this figure here, it's just showing the four different types of tertiary structure. We have a disulfide bridge between two cysteine residues. Then we have a salt bridge between a carboxylate and a positively charged amino group between aspartic acid and lysine. Then we have a hydrophobic interaction between these two aromatic rings on two phenylalanines. And then you see the hydrogen bond between these two serine residues here. All right, what kind of R group interaction might be expected if the following side chains were in close proximity? Well, if we take a look at this one here, this comes from a valine and this comes from a phenylalanine. Not that that's neither here nor there, but you can see these are both non-polar groups, so it's going to be hydrophobic interactions. So we'll just put here hydrophobic interactions. The next one's going to be a salt bridge because we have a negative charge here and a positive charge here, right? We have acidic side chain and then we have basic side chain. And so we're going to write here salt bridge. So salt bridge. And in the last one, it's going to be hydrogen bonding. Okay, so we'll scribble here H bonding, because we have an OH group here and the NH2 group. You could also have hydrogen bonding between the OH, right, like this, and then it could be between the oxygen of the carbonyl as well. All right, and that brings us to the quaternary structure of proteins, which is the final level, and it occurs when multiple tertiary structures, which we also call subunits, combine to form a larger protein. So these subunits are held together by the various attractive forces like salt bridges, disulfide bridges, hydrogen bonds, hydrophobic interactions. But each individual subunit has its own primary, secondary, and tertiary structure. And so an example of a protein that exhibits quaternary structure would be hemoglobin. It's made up of four individual subunits. You've got two identical alpha chains, which consist of 141 amino acid residues. And then you have two identical beta chains, and each of those consist of 146 amino acid residues. And each of the four subunits comes together to form the heme group, which is an iron atom sandwiched between the two hydrocarbon rings. And there's a picture of that in your textbook. Anyhow, so the four subunits are held together by hydrophobic interactions, creating an overall structure that's almost spherical. And you can see the structure shown here. So if you look closely, we have the two beta subunits that are highlighted in orange, and then we have the two alpha subunits here that are highlighted in green. And then we have the heme molecules here. So if I use my blue pen, there's a heme molecule here, 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 and here. And that purple sphere inside each one of those is an iron 2 plus ion, and then the oxygen binds to the iron, and then it gets carried throughout the circulatory system. Now, although hemoglobin's quaternary structure consists of those four individual subunits, it's not considered to be a large protein. It's got a molecular weight of 65,000 grams per mole, which is pretty big, and 574 rather individual amino acid residues. There's many proteins that are smaller in size, like insulin we saw in the last video, it had 51 amino acids in it, or much larger in size, like myosin has 6,100 amino acids. So a greater appreciation for the immense size of these proteins, even the small ones, can be achieved when you compare them to a common carbohydrate like glucose, because glucose only has a molar mass of 180 grams per mole and a molecular formula of C6H12O6, whereas the molecular formula for hemoglobin, get ready for this, okay? Its molecular formula is carbon 2,952, hydrogen 4,664, oxygen 832, nitrogen 812. I'm running out of space here. Sulfur 8, and then iron 4. So pretty big molecular formula there for your hemoglobin molecule. All right, here in figure 12.16, you see the structure of the human sorbitol dehydrogenase tetramer. And sorbitol dehydrogenase is responsible for converting sorbitol, which is an alcohol form of glucose, to fructose. The primary structure of each of the four subunits consists of 356 amino acid residues. The secondary structure consists of 14 alpha helices and 23 beta pleated sheets. The bulky and basically spherical tertiary structure forms when the alpha helices and the beta pleated sheets fold back on themselves, which allows the side chains, meaning the R groups, to interact through their attractive forces like the ionic bridges or salt bridges or disulfide bonds. And the complete sorbitol dehydrogenase enzyme structure is formed when four identical tertiary subunits interact with each other through hydrogen bonding, ionic bonds, and hydrophobic interactions. Now, sorbitol dehydrogenase is classified as a globular protein because of its overall spherical tertiary structure. And globular proteins tend to form stable suspensions in water or to be soluble in water because their nonpolar side chains are concentrated at the center of the spherical structure, whereas the polar side chains are concentrated on the surface, and so they can interact with the water, which is polar. Now, this figure here, 12.17, summarizes the key points about the four different levels of protein structure. That is, primary structure is the linear chain of amino acid residues. So it's the individual amide bonds or peptide bonds that hold them together. And those linear chains coil to form alpha helices or beta pleated sheets, each of which is considered to be a secondary structure. And those are stabilized by hydrogen bonding. Then, once you have the secondary structural motifs established, the protein can fold even further to produce tertiary structure, which is characterized by interactions between R groups, meaning hydrophobic interactions, salt bridges, disulfide bonds, and to a lesser extent, more hydrogen bonds. Then, if the protein is a monomer, meaning a single subunit, then it basically is good the way it is. But if it consists of multiple subunits like hemoglobin, then the subunits are going to interact via hydrophobic forces, salt bridges, and hydrogen bonding to form a unique quaternary structure. Okay, in what level of protein structure would you find the following features? So we've got an alpha helix. Well, an alpha helix is a type of primary structure. So alpha helix and beta pleated sheet. I know it keeps saying beta sheet in the textbook, but I call it a beta pleated sheet. That's what I learned. Okay, a salt bridge. A salt bridge that could be either tertiary or quaternary structure. Structure. All right. And then a peptide bond. Well, that's primary, right? That's the definition of primary. Primary structure. There you go.